Good morning. Yeah, thank you very much for that. I'm Dave Chapman, and I'm here to just speak for a few minutes about something called the Real Organic Project, which is a national effort, but it actually began here in Vermont. And it's pretty connected to everything that's happening at Nourish Vermont. Because as we become more sophisticated in our understanding of our interaction with the world and with food and how it's grown and how that affects us, we also suddenly are thrown into an arena where we're connected with how do we identify that food and how do we find it, how do we create connections between farmers and eaters. And the slogan that is very popular right now is "Know Your Farmer," and that is the best thing that we can all do. But all of us find ourselves in the store sometimes buying food that we don't know the farmer, and our parents and our kids and our cousins are all doing the same thing. So the Real Organic Project started in January, and it was originally intended as a Vermont effort to create honest labeling. And what we had discovered was that the National Organic Program has been slipping badly. And the integrity and the transparency of that label was slipping, was faltering. So I got into this about 5 years ago with my friend Davey Miskell, whom many of you know. I'm a farmer on the other side of the state, Longwind Farm, and Davey's right here in Shelburne, in Charlotte. And we found that the National Organic Label was certifying a lot of hydroponic production as organic. That's food grown without soil. It was certifying a lot of CAFO production. That's livestock grown in feedlots. And the things that organic meant to us were being lost. I had a conversation with a large hydroponic grower, nice man, and he was getting a lot of hydroponic stuff certified, and I said, well, you know, I'm on the other side on this. He said, no, I get it, Dave. You're real organic. And I thought, well, how do we take this conversation beyond the two of us to all of us about what does real organic mean and why is it important? And I think Zach's going to talk about why it's important. So I'm just here a little bit to talk about how do we bring some of that integrity back to the system so that all of us can go in the store and find the food that we want to buy. And I think right now that's not so easy. And there's also a process that the Real Organic Project is going to be working on to try and create more sophisticated conversations about this. Organic farming is not a lifestyle decision. You know, it's a decision about how we live and how we interact with the natural world. One friend of mine, a soil scientist, said in the 60 or so years since the organic movement began, as formed by Albert Howard in England, almost all the soil science, is amazing, almost all the soil science embraced and supported Howard's observations. So science has said, yes, he had it right, that biological diversity of the soil does result in greater health for the plants, for the animals, and for the people who are also part of that same system. despite that, what we see is a loss of that integrity because there's money involved. And we need to figure out how to deal with that. And I think that that's a political decision. It's all of us having conversations. So we began the Real Organic Project. It's got a pilot project going on this year. It's national. We have very well-respected people involved from all over the country. And it's generously supported by the Latner Foundation, among many others. So I hope you'll keep your eyes and ears open for it. We have a little table out back. From the rallies we had, some of you know about our rallies. Those were before the Real Organic Project, when we were still trying to reform the USDA. We have sort of set that aside and said we need to do it ourselves. And that's what we're doing. Okay. So now the real reason you came, and the reason I came too, is to hear Zach. He's doing tremendous work about, you know, what is it that makes us healthy, and what is health, and does it matter how the food is grown? And I'm looking forward to the answer to some of these questions. Okay, thank you.
All right. Thank you guys very much for all being here. This is one of the most beautiful spots on the planet, and I had no idea. It was a real joy to fly into this city yesterday, and of all of the approaches you do in an airplane, this is a special one. Incredible to see New York City from 30,000 feet and take a look at that, and then you see this lake and the pristine nature that surrounds that lake, and then sprawling solar panel fields scattered around the lake. It gives me hope. It gives me a real sense of joy. And then to walk into a room I've never been in and see familiar faces. You guys have followed me around the country, and some of you have been a real ministry to me as well, and it's a ministry of hope, because there's no shortage of need for that hope, as we are humans suffering in an isolated state of being, and then an overwhelming joy of walking in a room like this. And then what about Zekiah and what we heard this morning, that we are in church, we're in synagogue, we're in temple this morning together, and the power that we have today is extraordinary, and it gives me such joy to be present with you, to start to increase the gravity of change on the planet. And we're going to just do the quick disclosures just so that we get the CME stuff out of the way. I don't know why there's a gray box in the middle of that thing, but I'm founder and CEO of Biomic Sciences and Restore, the dietary supplement product line. Founder, the president of my clinic. My main disclaimer is I want to introduce these guys because I'm sharing a lot of science with you. John Gilday is sitting down at the microscope there. He is really the bulk of the genius behind all of our science that we do every day in my laboratories. Standing up next to him in the light blue shirt is Luis Montevelli. John Gilday trained at Hopkins. He was the first, among the first guys in the world to decode the genome of the Drosophila, which is the fruit fly, which will come up a little bit in our talk today. Brilliant man, and is on the cutting edge of the new genomics that we're going to talk about today. And then Luis Montevelli, standing next to him, is a brilliant scientist and physician from Brazil. He's an MD with a background in nephrology or kidney science, and a cell biologist. Gary Samuelson's in the purple shirt there. Many of you have probably heard of him or know him through the ASEA product line. He's an atomic physicist and always a joy to work around, always has some cool new toy in the lab for us to mess around with in the physics realm. And then David Roberts, standing there by the microscope in the dark blue, a master's in public health from Hopkins, and has helped a lot with our publications and all of that. So huge appreciation of that science team. And then William Vitalis, I'd be remiss not to mention him. William and I were the ones that really started down this journey of starting to understand soil and soil science. We call him the unicorn because every time we need a little magic, this is the guy that shows up with something. And so he's the director of esoteric sciences at my company, and I think he has the coolest title. So why are we here? We're here because we have a problem, which also means we have a huge opportunity. The problem that we face is that we've never been sicker as a population. If you've heard my talks before, there's a change on the slide, because the very first number on that slide as of a few months ago was 1 in 48. We're now at 1 in 36 children with autism in the United States. And so that's an extraordinary number because we're starting to approach the size of a kindergarten class, right? And so at 1 in 36, you're starting to approach a number of, there's an autistic child for every single kindergarten in the country now. And that's an extraordinary number because when I grew up in elementary school and middle school, and you can think back to your own journey on that, we had a disabled classroom, and it usually had 4 kids in it. And 2 of those 4 at least just had Down syndrome. We didn't know of autism. We never saw autism in the 1960s and 70s. And so the numbers were very low. We had 1 in 5,000 children with autism in 1975. And that number stayed pretty low all the way through the 80s, but then in the 90s suddenly started to pick up there. In the 1990s, we see this real steady rise starting to happen in the autism syndrome. And there was suspicion initially that we were just starting to recognize this new condition, and so we were maybe looking for it more aggressively, and maybe that's why we were starting to find it. But we had already reached 1 in 500 kids, so a tenfold increase from 1 in 5,000 to 1 in 500 by the mid-1990s. And then things got really wild. By 2001, we were at 1 in 250. And then by 2012 there, you're seeing 1 in 88 children. And at this point, we really knew we had a problem because we were starting to see an exponential curve. And this is when we started to really look more carefully at vaccines. We started to believe parents who were saying they had vaccine-injured children, and we started to ask, is there a correlation between allopathic medicine and our public health mission to children and the outcomes we were seeing? And that was way beyond the concern for vaccines. Everything we do in pediatrics. Pediatrics is obviously the highest antibiotic exposure of any patient population. If a kid goes to the pediatrician, you expect them to walk out with an antibiotic prescription, right? And so there's this journey down the medicalization of childhood that was seeming to correlate with this syndrome. And then things really got frightening as we've moved forward, because between 2012 and 2015, we doubled our rates. And we suddenly were on a vertical steep climb here of autism. And there had been some debate as to whether our criteria of diagnostics had changed from 1975 to 2005, and that's why we were starting to see it more often. But then with a doubling in 3 years, we suddenly realized, no, there's no way to fake the data here. We are just on a steady epidemic of neurologic damage in our children. Attention deficit disorder, 1 in 8 now are diagnosed with attention deficit or hyperactivity disorder. 70% of those that are diagnosed are medicated. And this is concerning because that what we medicate these children with is Adderall or Ritalin. These are equivalent of speed. And so you're putting these children on adrenaline to help them focus. And of course, that's what adrenaline does, right? When some threat jumps out at you or a car swerves in front of you, you want to be focused and be able to have a reaction that's appropriate to that circumstance. The downside of adrenaline and focus is the loss of creativity. You shut down the creative portions of your brain during an acute fight or flight state for all of the right reasons. When under attack, when a car swerves in front of you, it's a horrible moment to suddenly compose a poem to your lover and kind of think through, oh, is that a soliloquy or is that more of a, no, you want to just swerve and get safe and then have some peace, let the adrenaline slow down, and then you can go start thinking about the love letter again. And so that journey that we have into medicalizing childhood, adrenalizing our children so that they can focus on a multiple choice test, is starting to bring into question, what are we going to look like as humans if we are growing up generations of children that are drugged into a state of inability to be creative in their thought processes? And I think we can already see the answer to that question of what is it going to look like. We are going to be glued to a drug-like environment of input. We're looking for stimulation all the time, and stimulation that's predictable. When you are on adrenaline, when you are stressed, what you are looking for is predictability. And so the predictability of being able to push the same button on your Facebook feed to get the updates and know that you're going to get some input from the environment that is safe for you to receive, there's going to be no threat for you, but you've got some sort of human input, is a perfect drug. And so when you go to, a modern, school environment, you see kids that are literally talking to each other on their cell phones, right? They're texting back and forth when they're standing 5 feet from each other. And so we've gotten our children into a state of overwhelm where they feel safest in this very predictable interaction with the technology that's around them. So that's kind of the brain of the child. Of course, the numbers are probably far worse than 1 in 8. If you've been in a classroom recently, you know that far more than 1 in 8 children has a hyperactivity disorder. It's probably more like 1 in 4 that's exhibiting those symptoms. Asthma is now diagnosed in 1 in 10 children, again, underdiagnosed, but 1 in 10 are formally diagnosed. Allergies are both food allergies and environmental sensitivities are at 1 in 4. We have diabetes and prediabetes in our children, 1 in 4, obesity, 1 in 3 of our adults, and now in the adult state, major depression is hit 1 in 2. That's an extraordinary number in the adult because Freud and his colleagues around the early 1900s were really the beginning of evidence-based medicine and population health. And so they were studying really for the first time the exquisite public health numbers around mental health disorders. And they found that both in Europe and the United States, there was a rate of major depression of about 1 in 100. And you think back to the 1920s when they came up with those numbers, we had just survived World War I. We had just had the biggest flu pandemic on Earth, killed 20 million people in 2 flu seasons. We had, just an extreme stress of the depression happening in the United States. We think that our farmers are under stress right now, and I certainly wouldn't downplay that. Being on the farms today is a stressful financial environment, but it pales in comparison to what was happening in the farming environment as we headed in the Dust Bowl. And so, we were in this desperate state as a nation, and yet only 1 in 100 had major depression. Our brains were very resilient. We were very resilient to the challenges and stressors that were coming at us, and we've suddenly lost that capacity for resilience in the mind. Cancer just hit 1 in 2 in the American adult male. That number is astounding. That number was 1 in 9 as of just 20 years ago. And so to see these numbers climbing so dramatically is astounding in the neurologic conditions and the metabolic conditions. But cancer is a weird one, right? Because cancer takes some 20 years to form in a human body. And so just in a short 20 years to see a condition that's that slow at the biologic level suddenly go epidemic is highly concerning. And this is the cancer curves here. And so that's just between 1990 and 2015. In 15 years, we doubled our rates of cancer in the United States. And so there is something that got put in the water, it looks like, right? It looks like there is some vast exposure that's happening to our children and adults simultaneously that's damaging everything from our neurologic capacity to our very capacity to have self-identity, which is the opposite of cancer. Cancer is the loss of self-identity at the cell level. And then the dementia piece, an extraordinary number here at 100%. Big neuro study came out of the University of Virginia when I was there in around 2005. And it was a big population study looking at ages like 18 all the way to 95 and doing big neurocognitive studies in each of these individuals around the country. It was over 1,200 people were enrolled. And they found that by age 28, the entire cohort was showing signs of early dementia by age 28. And so we are sliding very quickly into memory processing deficits. I'm sure none of you are experiencing that, but apparently a lot of the population is experiencing, they set something down and they can't remember where they just said it, they are told something and then minutes later like, what the heck was that? It was so cool, and they can't place that one thing that was important. And so these are numbers that are just extraordinary, and the age at which these numbers are hitting us is extraordinarily frightening. In 1965, 4% of the US population had a chronic disease. Now, if we look today just in our children in 2015, 46% of our children have a chronic disease diagnosis. From 4% of the entire population to 46% of our children in that short period of 60 years and so, or 50 years there. And so we've got this amazing march of disease in our population, and it doesn't seem to affect all of us the same way. This is the curve for Parkinson's. And so this plays into this neurodegenerative dementia number. If 100% of us are showing some brain damage in the neurologic environment of memory by the age of 28, what's happening? This is looking at 1975 to 2005, and this is the trajectory of Parkinson's disease. And in the blue, you see the male. Down here in the orangish color is the female numbers. And you'll see that there's not a statistically significant climb in the women in Parkinson's disease over that time, and yet you've got this linear increase in males over that period of time. And then if we put next to that the numbers for Alzheimer's dementia, you see the opposite, where the males have not had a significant increase in the dementia due to Alzheimer's, but the women have had this steady increase over the same period of time. And so whether it's from Parkinsonian dementia or an Alzheimer's dementia, we're neurologically damaging our brains in different ways, depending on our gender, depending on our environment. The factor that kind of ties it all together is proteins. And so there's something called amyloid, which is misfolded protein that happens in the brains of individuals with any form of dementia. And so if you look at Parkinsonian brains, if you look at Alzheimer's brains, they have these big tangles of protein, tau proteins, or these beta-pleated amyloid beta plaques that form within the brain. It's not just the dementia, though, that does that. We see these same plaques and buildup in the pancreas of type 1 diabetics. We see them in the liver and pancreas of type 2 diabetics. We see this in patients with lymphomas and leukemias of today. Lymphomas and leukemias have changed a lot over the last 30 years. The type of lymphomas and leukemias we're getting is much different, and this phenomenon of protein misfolding as being part of that has really changed the environment. So we're seeing misfolding of proteins. Proteins, of course, are the building blocks of the human body. We have over 200,000 different proteins that build the human body, and there's 26 of these proteins that tend to really be prone to this misfolding phenomenon. And so we'll come back to that. So if we've got the neurologic system suffering, and now let's take a look at the bacterial environment and start to tie in the microbiome to this story. Cancer I'm going to focus on for a moment because cancer is the end stage of inflammation. So chronic inflammation is kind of the root of all of your chronic diseases out there. And cancer is kind of your penultimate state of so chronically inflamed that you've got a cell that's become so lost and isolated that it's now proliferating, trying to preserve life. It's under the impression it's now the last living thing in your body, and it's just got drive for life. And it's too damaged to differentiate, it's too damaged to actually repair itself. And a cancer cell is the most damaged cell in your body, which is interesting. A cancer cell, on average, has 20,000 to 25,000 unrepaired genomic injuries within the genes that make the proteins of your body. So it's a highly damaged, highly vulnerable cell that only knows how to now replicate as its only methodology for preserving life. It's interesting that a cell, all the way down at that microscopic level, has a drive for life. And I find that compelling. We are made from the fabric of drive for life. And I've seen that again and again. It's actually what pulled me into medicine, was watching babies being born in the Philippines. I was going into engineering, took a year off, went over to the Philippines, worked with a midwifery clinic, birthing babies. And the first baby that I birthed alone, I didn't mean to. It was in the back of a van. We were rushing to the hospital, and I was the only dude in the back, and this lady delivered her baby into the hands of a 19-year-old completely terrified guy. And at that moment, I saw just magic and miracle happen. This woman was very neurologically damaged from birth. She had beyond cerebral palsy. She was nonverbal, and she had been molested in the squats. She lived in the squats, just the slums of outside of the Philippines, outside of Manila. She'd been raped and had become pregnant through that, and she didn't even know she was pregnant. She had no cognitive capacity to understand the concept of pregnancy or know she was pregnant. She had just been dumped on the doorstep of the house I was living in. My aunt was a midwife, and so somebody just rang the doorbell at 3 o'clock in the morning, and we walked out, and this woman was hemorrhaging on the doorstep and was having what looked like a miscarriage. And so we stuck her in the van, and we didn't know her from anything, and she was clearly bleeding out, and so we just started heading for the hospital. And on the way there, she didn't look that pregnant, and so we weren't expecting her to actually have a pregnancy successfully deliver. And she delivered this baby into my hands that was fit into one hand. The child was so small, I had a little butt in the palm of my hand and the head on the tips of my fingers. and it was blue. It was this tiny little blue being that was so perfectly formed. And it was so miraculous as a 19-year-old who'd never seen miracles before to hold that little potential life in my hands. And it was sheer terror and joy when within a couple of maybe 2 or 3 minutes of thinking that this was a stillborn child, that thing started to breathe, started to pink up. And by the time we hit the hospital, it was the tiniest little screams you've ever heard in your life. It sounded like a baby mouse or something in distress. And this thing was screaming. And it still remains one of the loudest voices of my life. Just the injustice that everything about that moment had in it. It was unjust that the woman was damaged. It was unjust that she lived in poverty and squalor. It was unjust that there was no support system for her. It was unjust that she didn't know she was pregnant and raped. It was unjust that this child was probably going to die within moments or days of birth. So much injustice that child's scream kind of embodies for me. And yet there was a drive for life in that little baby. Why the heck would that soul want to be coming through that channel? And I believe there's an ancient soul in every one of you that's picked your pathway. And that soul showed up to be here for a few days, to scream into the atmosphere of Mother Earth, to say, I'm here. I want humanity to rise. I'm expressing myself in the terror of what we can create as humans in an effort to make it rise. There's something to this. There's something to this drive for life at the fabric level. That was a huge aside. I didn't mean to do that. Let's pull this back to the slide that's actually on the screen here. Sometimes I get into that moment and get lost. So there's a drive for life at the fabric level, and my background is in cancer research, and so I was an endocrinologist, internal medicine, then endocrinology and metabolism were my subspecialties in medicine, and then went on to hospice and palliative care later. But in the interim between endocrinology, or during my endocrinology stuff, I got into cancer research and was developing chemotherapy. And in that journey of kind of studying cancer under the microscope, came to really appreciate just how tenacious life is in a cancer cell and how hard it is to shut that down. There's just this drive that's within those cancer cells, and the drive for life there looks to have something to do with bacteria. And when I first heard these articles coming out of UCSF and UCSD, I just felt like, those are a couple of hippies out in California that are trying to tie the bacterial biome to cancer, when my lab at the University of Virginia knew damn well that it had nothing to do with bacteria, and it had to do with human genes as to why cancer forms, and that's what the dogma was at the time. But these started to come out more and more rapidly because we were starting to decode the genome of the microbiome in the late 2000s, and by the time we hit like 2010, we were starting to realize there is something very, very real about a change in the microbiome in the gut will predispose a woman to colon cancer. A different change in the microbiome will predispose her to breast cancer. A different one will predispose that guy to prostate cancer. And so we were starting to have this undeniable correlation between the bacteria that were living on the human being and the diseases that they would ultimately manifest, and that there was some sort of connection. Was too strange at the time. Now you fast forward just 4 more years, this is 2014, and it gets bonkers. What they find is, this is a qualitative survey of the breast microbiota. This is the microbiome of breast tissue. Has anybody been told that there is a microbiome inside your human breast? No. Somebody has. Either he was in my lecture before or way ahead of the curve. The microbiome of the breast is a fascinating reality, and we can do genomic testing and look for the scan for DNA within any tissue now. And what they found is that in the breast cancer of all these women, they were finding enrichment in Methylobacterium radiotolerans, one single species of bacteria out of the 40,000 that we know. you find one species that's enriched in every breast cancer tumor that you find. And so the theory was, oh my gosh, I wonder if it's an infectious disease. I wonder if the bacteria are infecting the breast, and that sets up a chronic inflammatory state, and eventually you develop cancer. This study is particularly cool because they went on to biopsy the healthy breast in the same woman. And what they found was a totally different bacteria in the healthy breast called Sphingomonas. And Sphingomonas was enriched over in that healthy breast tissue, and that was an aha moment to say, there is bacteria in healthy breasts. It wasn't just Sphingomonas, there's lots of other bacteria there, but the dominant species was definitely this Sphingomonas. At this point, the theory was, oh, well, maybe Methylbacterium secretes some sort of inflammatory toxin that causes the cancer, and so a shift in the microbiome towards Methylbacterium will predispose to cancer. But then they did this. They went ahead and correlated the DNA load showing the amount of that Methylbacterium within the breast tumor and the state or aggressiveness of the cancer. And they found the opposite of what they expected. The more sterile that tumor got, the more rapidly it killed the woman. The less bacteria that was present in the breast, the more aggressive the tumor got and the faster it killed her. An extraordinary single study to show us that the microbiome is what's keeping you from cancer death. As you stress the system, and it always begins in the quantum physics realm, we are not really molecular beings, we are actually atomic beings, right? Every molecule is made up of billions of atoms. The atoms are made mostly of vacuum space, 99.999%. And so you're 99.999% vacuum space. And sometimes you feel like it, right? You're like, that's an interesting reality, that what we're seeing with this study and others is that you have to develop chaos in the electromagnetic field within the vacuum space that is the vast majority of you, that will start to develop stress that will first show up in the microbiome. You're going to force a shift in the microbiome through the stress environment that you're coping with. Turns out that breast cancer, ovarian cancer, uterine cancer, endometrial cancer, all of those cancers are on a single electrical meridian of the body. All of the gynecological cancers that have gone epidemic in women run on one single meridian, which is where we put maternal stress. Men and women can have maternal stress. We can all have kidney meridian stress. But if we take a look at the state of the world, aren't we in a state of maternal stress over our soil, over our animals, over our nuclear families that are suffering from 46% of our children now with a chronic disease, epidemic of brain cancer in children, the mother side of us, the maternal nurture side of us, the sentient, emotive side of us should be in a crisis state, and we certainly are manifesting cancers that suggest we are embodying that stress, which first goes on to create an acidic environment. And as acid builds in an environment, Sphingomonas starts to have a very difficult time being, because this is a very respiratory bacteria. Methylbacterium, on the other hand, is very good at dealing with an anoxic, lack of oxygen, acidic environment, and so it moves in to do damage control where there is a problem. And that woman is now at risk. But at risk over a period of time, you can imagine what's happening is she's getting other conditions other than this cancer that's starting to brew in the breast. She's starting to stress her immune system, and so she's getting urinary tract infections, or she's getting chronic bronchitis, and she's starting to be exposed to antibiotics. And as her doctors continue to pound a declining immune system with antibiotics, we keep sterilizing the tumor that is potential in her breast. And so it's this steady decline of the immune system and the interaction with an allopathic medical environment that keeps pounding everybody with antibiotics that will keep feeding into this environment of sterilization and lead to a very pathogenic process. All right. Before you can get a cancer cell to do its thing, though, if we shift away from the bacterium, we know that we do need genetic injury. We know that we need to screw up the genes of the human somehow to get that cancer cell to go. And so we're going to take a look now at the microbiome and the human DNA for a second and ask this cool question: is what does it mean to be human? if you go to the American Cancer Institute or any of the kind of inside-the-box AMA-type medical communities, you're going to find a definition similar to this. Cancer is the most common human genetic disease. Transition from a normal cell to a malignant cancer is driven by changes to a cell's DNA, also known as mutations. And so it's damage to this double helix strand that's so famous, and this DNA strand has two templates that are tied together. And so the template or blueprint for life is tied into these strands of nucleotides that stay inside the nucleus of each one of your cells. And that DNA can be damaged frequently. It's constantly under threat. You go out in the sunshine and you're going to do billions of base pairs, and DNA throughout your body is going to be damaged by the radiation from the sun. Or in your bedroom, the gamma radiation coming out of Earth from radon and other sources of geologic stuff is doing billions of base pair damage. So DNA damage is kind of a normal thing in the existence of human beings, and yet we see this sudden change in our cancer epidemic, suggesting that there's something beyond just injuring the DNA. The question is, why isn't it repairing? If we've always been exposed to damage to the DNA and we're always been apparently pretty good at dealing with that, what is it that is now blocking our ability to repair that? How are we making a genetic disease, one that is due to the injury of the genes and their inability to repair, how are we making that syndrome, that disease process, go vertical like it is currently going? So let's take a look at human DNA before we kind of try to answer that question. It is grandiose. I'm going to go ahead and get somebody else up here so you don't have to just look at me. Can I have a volunteer, somebody up front maybe, so you don't have to walk too far? You want to come up? That'd be awesome. What's your name?
Jen.
Jen. That's my wife's name. All right. So Jen here has an arm. If we stick your arm out to the side here, that's about 1 meter in length. And so 1 meter is a nice distance for us to kind of wrap our head around 3 feet of space. And so we've got from kind of the inside of her shoulder to the tip of the finger about 1 meter of space. And that's how long this DNA strand that is molecularly, it's tiny, tiny, tiny little string is going to reach that meter. And put your arm down, thank you. Jen has about 70 trillion cells that make that body of hers. 70 trillion is one of the few numbers that's slightly higher than our national debt. So if we gave $1 for every one of your cells, we would hit our current debt state in the United States, or our deficit spending. 70 trillion is a very hard number to wrap our minds around because we have 1 meter of DNA in every single cell. I want to help you kind of understand how grandiose Jen is. 70 trillion cells times 1 meter. How many times do you think we could wrap Jen around Mother Earth if we put all of her DNA from 70 trillion cells end to end? All the way around the Earth. Do you think we could get her around the Earth 1,000 times around Mother Earth? That would be crazy. Jen, 1,000 times around Mother Earth? 2 million. 2 million times her DNA will wrap around Mother Earth. 2 million times. I fly all the time. You get a sense of the size of the globe when you fly. It's massive, massive planet we live on, and she would wrap around the entire Earth 2 million times with 70 trillion cells that are working in concert with the purpose of embodying her soul, some energetic spirit that is here on purpose. If we really could see each other, when Jen walked in this morning, you all would have gotten on your knees in awe of this woman. This woman is extraordinary. It's pure mathematical magic that she can stand here today in her existence and in her purpose. Shortly after getting up off of our knees, when we faced one another for the first time we met, we would hug each other. We would have an inability to avoid an overwhelming love for another being as complex and beautifully knit together as a human being in our presence. And so let's do that for a moment. We're going to stand up, and you're going to hug somebody next to you, and we're going to continue. Thanks, Jen. I appreciate it. All right. Come on now. That's ridiculous. It's going to turn into a frickin' orgy out there in a couple seconds. Got to put the brakes on. We got a public health discussion here. Out of control. David and Trey and Tree and all the other ones that put this conference together, thank you. Thank you for the vision. We are nourished in Vermont today. 2 million times Jen wraps around Mother Earth. But the weird thing about her DNA, not unique to her, every human being is unique in that only 1% of her DNA is going to make a protein. And so as geneticists and brilliant scientists, we came to understand that 99% of the genome is junk. And that's actually what we call it. We call it junk DNA, the 99% of your genome. Because that's what you see out in nature, right? You look around out in nature, it's just like, there's junk everywhere. It's just like waste, junk. Oh, a little bit of life. No. How is it possible that we've excused 99% of the genome and said, well, it doesn't seem to make a protein, it must do nothing? You can read articles from the leading genetics labs out of Yale and Harvard this year, and these two guys actually drive me more crazy than any other two human beings on the planet, but they will both, almost in the same language, say that the 99% of waste that you see in the genome, the junk DNA, is a leftover process from us emerging from single-cell organisms and slowly evolutionizing to the multicellular 50 trillion, 70 trillion. And so along that journey, we must have accumulated a bunch of baggage and waste that we just carry along. And it's the 1% that makes us this magical human being. It's such a narcissistic approach to understanding life when we start to see ourselves as, well, we're so grand that we have just dragged the rest of biology slowly up from the ground to become these incredible human beings, and the rest is waste. The 1% of the DNA was a really frustratingly low number because it turns out that only represents 20,000 genes. This number was very, very small compared to what we expected, because we already knew that there was over 200,000 proteins to make up a human body, and the model that existed in the 1990s was one gene would create one messenger RNA that would leave the nucleus and go into the cytoplasm of the cell and convert to one protein that would then know how to fold into an enzyme or into a structural protein and go and intercalate somewhere into the cell system to do its purpose. One gene, one protein, go do your work. Suddenly we found out we only had 20,000 genes. Not only was this dismal because we had 200,000 proteins and we now didn't know where our proteins were even coming from, more importantly, this number really was dismal because we had already figured out that Drosophila. John Gilday had already determined that there was 13,000 genes in a fruit fly. You're only 50% more complicated than a fruit fly? That seemed frustrating. But then within a couple years, they decoded the flea. Well, that doesn't sound terribly interesting until they found out that the flea has 30,000 genes. You are 2/3 as complicated as a flea. And that's reassuring. This is why you can't find your keys. This is why you can't, you're barely getting through the day, and you can write yourself a huge permission slip at the end of the day. 2/3 as complicated as a flea, doing pretty good. Got my emails checked. Boss thinks I'm clever. I'm good. Dismally low number, 20,000 human genes. How is that possible? What the heck is the other 99% doing? And what does it mean to be human again? So we are one species. We have 70 trillion cells. Only 20,000 genes. We don't know how that makes a human body yet. We're confused as to what that journey must look like, because it doesn't make sense that a template of just 20,000 blueprints could make over 200,000 different houses. The mitochondria are non-human, it turns out, and they live inside of our cells, and we inherit this. I call this the second conception. So I've developed this theory of the first, second, and third conception. And I think by the end of the journey, you're going to be convinced this is probably true. So the second conception, we need more genetic information. We need more information getting inside the cell than we can produce from our system. And so we have borrowed life from the mitochondria. Mitochondria look like bacteria, except their DNA looks more like a virus. And so bacteria look like they're the offspring of viruses interacting with bacteria early in the journey of creation. And so a virus infects a bacteria and it turns it into this strange organism that's able to convert fat or sugars into a single molecule called ATP, adenosine triphosphate. That molecule is the only fuel the human cell can run on. And so bizarre that to become even that first sperm or the first ovum, you need energy that's being produced from a non-human source. It turns out that all of your mitochondria are inherited from mom. So with your first conception, you get 23 chromosomes from dad, 23 chromosomes from mom. And at that point, dad's kind of done with the process. He bought the six-pack to get the whole thing started, showed up for a few minutes, and then mom took over with the magic of creation. And she donated mitochondria through her ovum, through her egg, to what will become a fetus. There's 3 species of mitochondria, interestingly, and there's not enough data yet as to why there are 3 species. And I think we're going to find out all kinds of cool things about that in the future. But there is a lot of these guys in the human body. So it turns out we have 14 quadrillion mitochondria in a gen. Okay, 14 quadrillion. This is a number that's very difficult to wrap your head around. Not too many genes, 37 genes, but when you multiply those 37 genes out by 14 quadrillion, you've got a ton of genomic information, and there's junk DNA in there. There's non-coding DNA within that strand as well. And so this is our second inheritance of genomic information is from the mitochondria. Then we get into the bacteria. Once again, this should be inherited. This conception should occur in the birth canal of mom's vaginal canal, right? Well, that's right, unless you're born by C-section, in which case you never see mom's vaginal canal. But in that journey, there's a great TED talk on the microbiome of the vaginal canal and the genome of the infant as it changes from newborn to about 6 months of age. The microbiome changes radically over that time because at birth, the child should look exactly like the vaginal flora of mom, which actually looks a lot like the bowel flora of mom. And so you have a lot of very specific bacteria there, but you also have a ton of diversity of bacteria, fungi, and other species that are there in the vaginal canal. Take a look at how many genes the bacterial species can contribute. Somewhere around 2 million genes have already been mapped out in this bacterial environment. And so our 20,000 genes suddenly logarithmically outnumbered by the microbial genomic information that would be programming which proteins are converted in our body, which are produced, which proteins go into a useful state, etc. So we've got one species, 20,000 genes. We've got the 40,000 species of bacteria with 2 million genes. And then we get into the stuff we don't think about. Because when I say microbiome, whether you're a doctor or not, your first thought is going to tie to bacteria. And this is where we've kind of really fallen on our face again with missing the forest for the trees, is the bacteria are just the very, very beginning of it. There are 300,000 species of parasites. 300,000 species of parasites. About 6 or 10 of those have been found to be pathogenic, meaning that if they're in your water, you could get a diarrheal illness and have some inflammation for a while. The other 294,000 or whatever, it's just an insane number of these parasites that don't seem to cause us any problem. In fact, there's a realization now that the parasite that lives under your eyelid, I just said that correctly, there's a parasite living under your eyelid right now if your eye is healthy. If you lose that parasite, it looks like a little worm under the microscope. If you lose the worm under your eye, you suddenly become very prone to conjunctivitis, irritation of the cornea, and glaucoma if you screw up the microbiome of the eye specific to the parasites that should be living there. 2 billion genes in those 300 species of parasites. 2 billion genes. There are 5 million species of fungi. 5 million species, not genes, species. 5 million species of fungi on planet Earth around us. We think of the shiitake and the oyster and some of these mushrooms. We're like, wow, look at our grocery store's awesome. We have like 7 mushrooms. 5 million species of fungi on planet Earth. This true trillion genes just got revised because we're just starting to decode the genome of the fungi. It's now at 125 trillion. 125 trillion genes in the fungal kingdom. We are a drop in the bucket of the biology on planet Earth. Finally, the viruses. We have no clue how many species of viruses are on planet Earth today because there are too many of them to begin to characterize. We do have a good sense of how many viruses are on Earth. The number seems to be somewhere around 10 to the 31. That is a 1 with 31 zeros after it. We don't have a name for that number. 10 to the 31 is 10 million times more viruses than our stars in the entire universe. Stars in the entire universe. There is a virus for every one of those stars times 10 million on planet Earth today. And we have a public health mission to get everybody a flu vaccine. Well, thank God for allopathic medicine, because what will we do without the flu vaccine? I mean, it's the most ludicrous thing on the planet that we can excuse the science behind it and then excuse the public health science behind it. The flu vaccine is one species with a few variants that we're vaccinating ourselves against. Never mind that the NIH's own study looking at the efficacy of the flu vaccine has shown year to year zero efficacy of the flu vaccine, with the exception of one little category. It's useless in children, it's useless in anybody over the age of 55, but it's very helpful in males between the ages of 45 and 55. And we're not really sure why the specificity. Why do the males, when they're playing golf, benefit from a flu vaccine? There's probably an answer there waiting to be had that will be fascinating when it comes out, but right now we don't know. Why? What is the benefit to those guys? The only measurable benefit to the flu vaccine is that they have 6 hours less of symptoms when they get the flu. There is no change for any population in the rate of flu that happens within the population. The NIH scientist that chaired that committee to do that study was fired immediately upon publishing that study. We have a low tolerance for the exposure of failure. And that's just human. That's not a conspiracy theory. Everybody hates being told they're failing. And when we're putting billions of dollars of medical research and billions of dollars of drug development into our vaccine program, we sure the hell don't want to be wrong. We sure the hell don't want to fail, because people are dying of flu. Well, not very many, but people might die of flu, and we should be after that virus. And then we look at the genome and we think, what the hell are we doing with anything, talking about a vaccine to anything? And interestingly, I don't have this slide for you. I should have it in here for you, but take a look. You can probably just Google this. Google Images will probably bring this slide up for you very quickly. Google rate of autoimmune disease and vaccines. Ever since the very first vaccines for polio, then the measles, then the mumps, then rubella, and running through the rest of them, did you know we vaccinate our children to 64 bugs these days? Well, that makes sense because there's only a few trillion out there. But we picked the 64 most dangerous of the 60 trillion out there, and we vaccinate our children to these things. And every single vaccine that comes on the market correlates with a higher increase in autoimmune disease. We get a steep climb in the rate of autoimmune disease in the population as we vaccinate ourselves just against a few of these microbes. We're going to come back to that. Epigenetics. This had to be born. We had to birth a new field of science called epigenetics because we found out that 20,000 genes were making over 200,000 proteins, so it blew apart our model of how genes work. What time does this talk end? I know we got started a little late. What time do you want me to wrap up, just so I can keep an eye on that? Because I could go till sometime next Tuesday, so I need a stop point here. It's 10:30 now. It's 11 o'clock. 11:15. Do I hear noon? Do I hear noon? All right. If I don't have time for questions at the end, there's a booth that I didn't know I was going to have. So there's a table with a piece of tape that says Zach Bush on it. So I'm going to be selling myself out there. And so you can come to my Zach Bush table, and I can answer questions all day long for you. I'll try to take some questions at the end, but I do have a lot of information I want to share with you. And I always get sidetracked. All right, so 20,000 genes have to create 200,000. We had to create this new field of epigenetics. And epigenetics was the understanding that the DNA had to be somehow modified to get genes to turn on and off. But more important, we had to come up with an eloquent mechanism by which things could not just turn off a gene, but make it shift to build something different. And so epigenetics began with the understanding of methylation patterns on the DNA. So methylation is a mechanism by which we can turn on and off a gene. Methylation patterns were starting to be recognized that the environment, when you take a breath and you have mold toxin in that breath, it can methylate a bunch of very specific regions within your DNA to block the ability to make some proteins. So there's some mold toxins, Lyme toxin, there's a number of different things that have been shown to methylate, for example, the promoter region of the DNA strip that would go on to make the protein called glutathione synthase, which is the main machinery that makes glutathione, which is your primary antioxidant in your body. And so exposure to the environment, we were starting to realize, could turn on and off genes, which was totally different than what we had thought just a few years earlier in the mid-1990s. And so we were starting to realize we were very plastic. Our genome was shifting every time we got exposed to the environment. Of course, with that much microbial life that I just showed you around us, it was starting to bring up the question, whether the microbiome is interacting in an epigenetic fashion with our genes to play havoc or play health into our system by manipulating the way in which our genes would turn on and off. But we had to go beyond just turning on and off genes with methylation patterns. We needed to figure out how a single gene could build 200 different proteins. Because we knew that was already happening. And so imagine a blueprint that you go build a house from. All right, so it shows you exactly which walls to build. It shows you how to put the house together. One blueprint equals one house. Imagine the blueprint that would allow you to build 200 different houses. That was what we were struggling with. How in the world does the nucleus within the cell figure out how to build those 200 different things that look completely different? And it's not like variations. It's not like, oh, there's an addition on this version of the house. 200 completely different houses are being built from one template. And that came down to this new field of microRNA that John Gilday has been right on the forefront of. And so the microRNA, it turns out, shockingly, are coming from the junk DNA. That 99% of your genome is not waste. It's making something. We didn't know what it was making, but of course it's doing something, because we already knew, and we weren't talking about the fact that the junk DNA is as carefully preserved as our mere 20,000 genes as we pass them on to our offspring. So we were carefully managing and correcting and passing on carefully the junk DNA to our offspring, because the 99%, of course, is going to control the 1%. So what is the 99% making? It's making something that we call now microRNA. Before this, we had messenger RNA, which would take a template from the nucleus, take it into the cytoplasm, and produce a protein. MicroRNA never make a protein. That's why we didn't find them. That's why we thought that the DNA was doing nothing, is it wasn't ever going to result in something that we could see under the microscope. Once we got down to the capacity to find these microRNA, we found out that these are very special because they can leave the cell. Messenger RNA stay in the cytoplasm of your cells. These guys are so tiny that they can leave the cell and traffic to distant cells within your bloodstream. Even more bizarre, these guys can actually get into vacuoles, these little envelopes in your breath and suspend in the air when you speak. They can get into your sweat. They can get into your urine. They can get into your fecal material. And you've got oozing from you genomic modulators. A single gene can produce over 200 different proteins depending on the co-activators and co-repressors segments of these microRNA. This means that your genes could produce over 4 million different variants of you. Your mere 20,000 genes could build 4 million different bodies today based on what kind of information you're getting in from the microRNA. And so there's something very cool, and I get a little bit narcissistic on this because it seems so powerful, but I have had an effect at the genomic level on every single one of you because I've been talking elevated over you for the last 45 minutes. My breath, for better or worse, is right now changing who you are. That's startling and very cool. Have you ever seen two people married for 80 years? Have you seen two people married for 40 years? Do they start to look like each other? Isn't it weird? My daughter isn't so sure about humans, but she is fanatical about dogs. Have you noticed somebody who's been living with a dog for 13 years? That's weird. I mean, that's a little creepy now, right? Pick your dog carefully and just look in the mirror and be like, now, if I were to start to look like that animal, is that a good thing? Or so, I've already got kind of a prominent nose, so I probably need like a bulldog or something, with not much nose going on there, right? And so pick your animals carefully, pick your spouses carefully. And I actually am very serious about that last piece. If you are frustrated about your health, you can build 4 million different bodies from the template you have right now. If you are trying to change, and you're changing your environment such that you will transform into a different body, and I see this happen regularly in clinic, right? You go 2 years into my clinic and you see the same population walk in 2 years later. They are different. They look different. They move different. They think different. They are different human beings. But there's a segment of them that are stuck, and they look the same. There's a segment of my patients that are stuck. They look just as sick as they did 2 years ago. And I'm trying to teach them how to change. I'm teaching them breath work, and I'm teaching them exercise work, and I'm teaching them everything I can think of to make them build a different body, and they're stuck. And I got very frustrated for years about this, and my patients were getting way more frustrated than I was because they were doing everything I told them. I was blaming them, well, you must not be compliant. And they're like, fuck you, doctor, I am compliant. It is very frustrating being a human being stuck. We don't like stuck. We are born to transform, which is cool. We're born to constantly be transforming and improving and getting stronger. And it's very freaking frustrating when you're stuck. And as I started studying the microRNA, I realized, oh my gosh, I've never met that woman's husband. And so I started to demand that my patients that were stuck bring in their family with them. And that changed the equation big time. I realized she was married to somebody who was stuck. She was married to somebody who would not change his diet, who would not change his behavior on Sundays, watching the game or whatever it was that would frustrate her and make her feel unloved and isolated. And she'd be stuck doing chores and taking care of the kids on Sunday afternoon while he's depressed about their financial situation and sitting on the couch and doing whatever he needs to do. And she's stuck because he's exuding in his sweat, in his urine, in his semen, and in the saliva when she kissed him in the morning, microRNA that was programming for the same freaking thing over and over and over again. You change a family, you see radical transformation. because the whole microRNA environment is shifting fast. Did you know that the animals are suffering the exact same rate of increase of cancer that we are? We are diseasifying the environment through our own stress. Our microRNA is programming the world for cancer. Our microRNA could program each other for joy, for transformation, for sheer human potential. MicroRNA travel far and vast, and they can change everything. An extraordinary look at the microRNA in the human bloodstream. Keep in mind that we're only about 4 years into our understanding of microRNA, and we're less than that of starting to decode what do the microRNA look like that are coming. What it's going to make, but it's telling you what the environment looks like. And so the microRNA, this one doesn't work either. I'm just going to come out and start hugging you all, and it'll be over, okay? All right. Test, test. You guys got me there? Something? All right. A little bit louder? All right. 15% of your microRNA from bacteria, 15% of your microRNA from the fungi, and 5% from food. The interesting thing about the microRNA is it's going to express exactly what the organism was feeling a few minutes earlier. And so it's very plastic. The microRNA right now that you're expressing from your junk DNA is a little bit inspired, I hope. I hope it's feeling a little bit of love from whoever you just hugged. It's expressing some good stuff right now. But when Monday comes around, you're back at work and you're stressed out, you're going to be producing totally different microRNA. Now, my company is developing some products for cattle and other feedlot animals to try to reduce the suffering in these organisms. So I've spent a lot of time on feedlots. And the first feedlot I went to, I was shocked because I recognized exactly what was going on. All of the cows had autism. They all looked just like the autistic children in my clinic. They behaved just like it. I'm from Virginia. It looks a lot like this. And we have these bucolic rolling hills with the cows grazing. And my daughter's favorite animal is the cow. And so we have to stop on the side. And this is when she was little. She's now 18 and doesn't even want to drive with me anymore. But yeah, so back in the day, we would stop and she would walk up to the fence and like 5 cows would come running over to see her, right? And they'd be blinking with those giant eyes, huge eyelashes. Beautiful spiritual moment between a little girl and a cow. Feedlot, they can't even make eye contact with you. In fact, as soon as you step into a pen, they run the other way and start hitting themselves against the cattle rails and everything else. They are as skittish and as terrified and overwhelmed as an autistic child. And I know moments before, just a few months earlier, they were a bucolic normal cow with no autistic behavior whatsoever on a farm somewhere, and then they get shoved onto a CAFO feedlot for the last few months of its life, and they all become this autistic, neurologically overwhelmed state, and they are in terror. And then you walk into a chicken farm. What the hell are we doing as human beings? How is it possible that we did this? These animals have their beaks clipped off so that they don't commit suicide, so they don't peck out their own hearts. We clip off their beaks and we put them in cages so they're so small they can't even turn around. A boiler chicken will go from chick coming out of the egg to on your plate in 6 weeks. What a brief life of misery. Hopelessness, terror, anxiety, and a sense of impending doom from the moment they're born. Guess what microRNA are turning on in that chicken? Terror, fear, a sense of impending doom, anxiety, a sense of nobody cares about me at all, and it ends up on a chicken salad somewhere. And as good doctors studying incredible nutrition, as we do, we have no idea, we tell the woman, you should just eat chicken salad probably. That would probably be good for you. Don't eat steak. No, I think chicken salad sounds pretty good. Why don't you try that for lunch? And inexplicably, 2 hours after lunch, she has an anxiety attack. Panic attack to beat the band. Driving down the road, no stress, hasn't had to pick up the kids from school yet. she suddenly has the overwhelming sense of impending doom. I am dying, and nobody cares. I'm unloved. And she comes and tells me this in clinic, and I'm like, well, it must have been a panic attack. And she's like, it doesn't make any sense. My husband loves me. I've got a great life. Don't have any financial stress going on. Like, why this? The why is starting to look like the food just made her have a panic attack, because the food she just consumed was in the midst of a panic attack throughout its existence, but certainly in the last few minutes of its life. You are what you eat has become extremely real statement. Extraordinary journey we are on as human beings in this experiment of torturing our food chain. Can you imagine being a piece of corn in the middle of a 10,000 acre genetically modified corn crop? Through the genetic modification, it's allowed to be sprayed with Roundup a few times in its lifespan, which depletes it of all of its nutrients that it would deliver to life beyond it. We have ripped out of the corn and the soybean on this planet its very sole purpose of passing on nutrient. Do you think that's depressed crop? I think so. I think at the energetic level, we are creating depressed plants as much as we are overwhelmed and anxious and abused animals. This is, in the end, your gene pool. This is exactly what makes you who you are today, is everything. We are starting to understand singularity at the atomic physics level. We're also starting to understand it at the biologic level. We are all going to express the same thing. We all live on a tiny, tiny little speck of blue in the middle of space. If we kill it all, we will die. It's that simple. We are killing the environment around us, and we are dying. Antibiotic usage. If I'm right in that the loss of bacteria, and if these other studies prove out that the more sterile the body gets, the more we die from cancer, then we should see a huge correlation between our antibiotic prescription behavior as physicians and the death from cancer. This is from 2013, looking at the antibiotic prescription behavior in these different states. The darker the state, the more antibiotics are prescribed. On this side, you see the redder the state, the higher the death toll from cancer. Is there a state-by-state correlation right there? State by state, the more antibiotics we prescribe, the more people die from cancer. The more cells get isolated, the more antibiotics they get, the more isolated they get, the more lonely they get, the more confused they get, the more cancer it gets, and then we sterilize that new cancer and with more antibiotics, and it becomes metastatic and will kill the person. It's new. We've never had this cancer pattern. This has never been the map of cancer in the United States. It's always looked like this, which showed us that living in Vermont is a really bad thing if you want to avoid death from colon cancer. Northeast predominance here, and that it was the map forever, but certainly from 1970 to 1994. This is the map for lung cancer. Again, north looking like a disaster area. Interestingly, prostate cancer, again northeast, but now pulling in this huge prevalence in the northwest. And so there's always been this northern predominance to cancer in the United States until the short 15-year span between 1994 and 2007 here. In that span, we suddenly see a reversal of the death of cancer in the United States of America. We are the most heterogeneous genomic population on Earth. We have people from all walks of life in the United States. We know that there should be some sort of genetic safety or predisposition towards cancer, and yet in this heterogeneous population that we are, in just 15 years, we completely, radically changed the public health statistics of death from cancer. That's impossible if we are right about what cancer is. It's impossible to describe that out, and yet there has not, even though these maps are from the CDC, Centers of Disease Control in Atlanta, I have not seen a single public announcement from them or warning from them that we have suddenly done something to the environment to radically change death from cancer. And in this way, I don't think the doctors are culpable because our antibiotic prescription behavior hasn't changed. This flat line from year to year is looking at the amount of antibiotics we prescribe. And I was very relieved when I found that graph. This is from a New England Journal article in 2010, or 2011 rather. And the study was showing this antibiotic prescription behavior in doctors. And amazingly, we prescribe a lot, 7.7 million pounds of antibiotic per year in the United States are prescribed by physicians. 7.7 million pounds of antibiotics are being prescribed to our men, women, and child, but it hasn't changed to radically excuse or explain the way in which this map has shifted. And so this is where we get into this molecule that you guys are all very aware of as a unique audience. Glyphosate is the active ingredient in Roundup. Glyphosate is patented as an antibiotic, antifungal, and antiparasite. Interestingly, it never got patented as a weed killer, but that's what we use it for. How does it kill the weeds? It destroys the building blocks of life within the soil, microbial life, as well as in the plant itself. And it does that through some unique pathways, and one of them is called the shikimate pathway. The shikimate pathway is a complex series of enzymes that is very unique to bacteria, fungi, but not in animals. And so neither the chicken, the cow, nor the human has the ability to produce the shikimate pathway or the products that the shikimate pathway would put out. What it makes, importantly in this world of genomes and proteins, is the aromatic amino acids, including phenylalanine, tyrosine, tryptophan, histidine, thymine, L-dopa, 5-HTP. You guys maybe have heard of some of these. But of those, the ones that's probably most important are the top few. We call these the essential amino acids. There's only 26 amino acids that build your entire body. It's very much like the letters of the alphabet. We have 26 letters in the English alphabet. We can build over 200,000 words with those 26 letters. And within the alphabet, we've got 8 vowels. And the vowels are kind of our most precious, conserved letters because every single word needs a vowel. Same with the essential amino acids. They're highly preserved and they're highly needed, and we can't make them. So of the 26, most of them we can make in the human cell. But the 9 that we cannot, we call the essential amino acids, and many of them are on this list, including phenylalanine, tyrosine, tryptophan. It turns out that the main precursor that tyrosine and tryptophan represent are the precursors to dopamine and serotonin. We can't make them. And yet the 2 largest populations of neurotransmitters that make your brain work and would prevent depression from happening and would prevent the decline in neurologic function in a child with autism is dependent on the enzyme pathway that is blocked by glyphosate. And so we have brilliantly, as human beings, put a water-soluble toxin into the planet that is 70% water that blocks the ability of microbial life within your gut as well as in the soil to make the building blocks for healthy proteins. And so let's take a look at the spraying map for glyphosate. If you've seen any of my talks, you've been bored with this slide, but this is 1992. We had to be very careful where we sprayed it in 1992 because it kills all the plants it touches. And so we had to spot spray the weeds around farmland and everything else. And of course, in the 1990s, we also got direct-to-consumer allowance from the EPA, actually in the 1980s. And so the homeowners started spraying Roundup everywhere. And this is a bigger problem because farmers are actually very conservative because it's actually expensive to treat an entire farm with Roundup. And so they'd be very conservative as to how much and where they're using that. You've seen the guys with the backpacks in the suburban neighborhoods. They are hosing down the driveways with glyphosate. Roundup. Roundup's not a treatment for pavement. It's not a treatment for concrete. It's a weed killer for the 6 dandelions that the guy is trying to kill. It happen to be anti-cancer foods. Amazing thing, dandelions. But we kill it with glyphosate because it really sucked to have to bend over and pick it out and make a salad. And so we kill it and we spray that and it washes down our driveways into our municipal water system. So we started drinking Roundup in the 1980s. Interesting experiment in the microbiome. By the 1990s, we were starting to spray crops. 1992, we started to spray crops. What crop would you want dead before you harvest it? Wheat. Oats. Exactly. And so you look at lentils, you look at some of the beans, you look at your oats, you look at your wheat. We started using Roundup not as a weed killer, we used it as a crop treatment called we called it a desiccant or drying agent. That's a pretty fancy word for death agent. We were killing the plant prematurely so we could harvest it earlier. So we were now delivering food to the consumer that was sprayed with glyphosate shortly before harvest, and we sprayed it early, which means that grain never got to mature to its full potential. What happens to grain as it naturally dries on the stalk? Well, lots happens. The proteins change, the genomics change of that plant. We started eating prematurely killed gluten and glyphosate-ridden molecules in our wheat in 1992. By 2011, we had the advent in 1996 of the genetically modified crops, allowing us to spray corn, soybean, sugar beets, some of the potatoes. And so now we're spraying all of our staple crops way beyond the grains with that chemical. I don't like poor correlations, and this looks like a poor correlation here with the cancer map. I wanted to see the highest concentrations of spraying right here. And so that's what I was after when I started trying to pull these maps together. And what we found, of course, is that the predominance is up north there. So some 85% is being sprayed in this region right here, which certainly there's a little echo of cancer heightening through that space, but not really the highest rates. And it wasn't until we overlaid this map that it all started to make sense. You can't see it probably well from where you're at, but in white we have the outline of the United States again, and in red, this is the tributaries of the Mississippi River. That huge torrent that sits over this entire region of the United States pulls all of that glyphosate, which is a water-soluble toxin again, into its waterways and delivers it down the last 90 miles of the Mississippi is where? Baton Rouge to New Orleans. Where's the highest cancer rates in the entire world now? Baton Rouge to New Orleans. We call it Cancer Alley. In 20 short years, we created the epicenter of cancer death in the world on the Mississippi River deltas with one molecule, glyphosate, Roundup, an extraordinary, insane journey towards cancer death. Lyme disease. You guys get a lot of story around Lyme disease up here. There's stories of, government laboratories and maybe militarization of the spirochete and some things, and then on Long Island it broke loose with the first things and kind of infected that. Something is changing in our Lyme pattern. Look at this. Huge cluster of Lyme right over our densest spraying areas of glyphosate. That's not what we used to see when we just thought this was a tick-borne disease. This is the rickettsial infections now in our country. These are other tick-borne organisms that move through our animal population, overlapping really well through that same whole region where we see cancer death. This is Ehrlichia, another infectious process from our animals that can infect humans, cause liver disease. Same patterns over and over again. We're creating a decimation of the immune system in this region of the planet, and that's leading to the antibiotic behavior of the physicians. They're coming in with bronchitis, they're coming in with urinary tract infections, they're coming in with all kinds of complaints of infection. Chronic Lyme is probably among the worst diagnoses you can get because you're going to be put on 6 years of antibiotics. I've got patients that have had 16 to 20 years of antibiotics from Lyme in Lyme clinics around the country. I've got a couple patients that have been on more than 6 years of intravenous antibiotics in the home for Lyme disease. It's criminal behavior from integrative medicine that we're giving this much antibiotic into the human population and sterilizing them. Those patients do horrible, by the way. You put anybody on a year of antibiotics, you just destroyed their next decade. It should really be illegal. Mental illness in the United States. And so it turns out that over this same period of time, we've seen this huge shift. Again, to reach the rates of major depression that we now have in the Mississippi River Delta, you have to remove sunshine completely from the human being. All right, so up here we get this huge seasonal affective disorder in our poor people from Portland and Seattle that I had breakfast with and everything else. It's that kind of environmental stress that you have to put the organism through to reach these levels of major depression. If you overlap antibiotics with this, it's very fascinating. I don't think I have the slide here. Let me check. Yeah, it's not here. But I've got another slide that I've done on this that's pretty fascinating, looking at antibiotic usage and the occurrence of major depression and anxiety disorder. One course of antibiotics, single course for 3 days for a urinary tract infection or 3 days of a Z-Pak for bronchitis, there is a 17% increase in risk of an anxiety disorder or panic attack happening in the next 12 months. There's a 24% increase in the risk of major depression happening within the next 12 months. 24% increase with one course of antibiotics. If you get 2 courses of antibiotics, 54% increase in major depression in the next 12 months. Has a doctor ever told you that? Handing you an antibiotic prescription? Oh, be aware, this is your second urinary tract infection in the last year. I'm probably increasing your risk of major depression by 54%. If you kill the microbiome, you will have massive dysfunction. Currently, glyphosate is our #1 chemical worldwide. We're spraying over 2 billion kilograms of glyphosate into the soils of the planet Earth right now. That's 4.5 billion pounds of an antibiotic being sprayed into our environment. What does it mean to be human? I made this slide last night for you because I was thinking a lot about this. I had a really stimulating conversation with some of you in the lobby last night. And this was kind of at the forefront of my mind. What does it mean to be human? So I Googled to be human. And for some reason, Wonder Woman is the very first hit. And I just thought that was weird. I was like, really? Like that? Wow. I've really underestimated my potential here. I should be a 6'1" supermodel who can do stunts out of bed in the morning. That's what it is to be human. The other human that happens to pop up, though, is Desmond Tutu. The other humans that pop up look like this. They're all from tribal environments. There's not a single healthy Caucasian on Google when you type, what is it to be human? I was stunned. There are pictures of Mark Zuckerberg looking like Data from Star Wars. They've made his face to look like Data. That's the closest thing you get is a cyborg version of Mark Zuckerberg when you say it is human. To look human on Google right now, you need to be from Africa. This is one of the other Caucasians, of course, not surprising, Arnold Schwarzenegger as cyborg from the future. It's an interesting Google search. We are losing our self-identity as a species. We're realizing that we are more computer than we are human in the developed world. And I think that's pretty fascinating. What does it mean to be human? What it means to be human, it has to have something to do with the microbiome. There must be something deeper than just cancer because we have many more epidemics than just cancer right now. And we're going to tie this back to chronic inflammation. I really am concerned about steamrolling somebody else's time here. It's 11 o'clock now, and I heard 11:30, 11:45. Are those numbers real, or is that just hopeful from this group? Is that somebody who, what do you guys, do I need to wrap up, or do I have another half hour? 20 minutes, okay. Another talk at 11:30? Okay. All right, so we need to tie all of this back to chronic inflammation, which we know is the root of all disease. Inflammation has to come from the immune system. The immune system lies mostly in your gut lining. We call it the GALT, the gastrointestinal-associated lymphoid tissue. 60 to 70% of the volume of your immune system there, but over 80% of the antibodies that are made in your total body lie there in the gut lining. These finger-like projections are called villi throughout your intestinal environment, and you see them, they are kind of coral reef-looking structures. They seem poorly engineered in that the lining, the epithelial lining or protective membrane that separates you from the outside world is only one cell layer thick. That's 50 microns. That's half the width of one of your hairs. So pluck a human hair, cut that in half longitudinally, and now that's the width of the cellophane-like layer that separates your immune system from the outside world. That's bizarre engineering. That cellophane will cover 2 tennis courts in surface area. Your skin only has 1.5 meters in surface area. 2 tennis courts of surface area is your gut lining, and it's only half the width of a human hair, 1 cell layer thick, and it's fully dependent not just on those cells, but it's actually reliant on the tight junctions or the Velcro that will tie those microscopic billions of cells into a carpet that would cover 2 tennis courts. And so all these tiny microscopic cells, trillions of them, have to line up to make this coherent, intelligent barrier system to keep the outside world out, absorb nutrients in an intelligent fashion, and keep the toxins and chemicals out of your body. That's the process of being human. It turns out that at the molecular level, to be human is to have an intact barrier so that you can say, here is me inside, everything outside of that is potential foreign invader, and I need to attack it. So your immune system is programmed to recognize self and to respond to non-self. The issue that we have at hand is that it's the same Velcro that ties the bloodstream together. So all of your blood vessels are tied together by the same Velcro proteins that hold your gut lining together. It turns out it's the same Velcro proteins that tie your entire blood-brain barrier together to protect your brain and peripheral nerves from whatever's in your bloodstream. It turns out that the same tight junction proteins, the Velcro, is holding together the tubules, the proximal renal tubules and beyond, that detox your body. And it turns out that glyphosate, after killing the microbiome in your gut and the soil, after blocking the ability of your microbiome and plants to make the building blocks for human health or a human body, it also goes on to destroy the Velcro. This is the small intestine growing under our microscopes at our labs. And so we have highlighted ZO1, which is an element of the tight junction Velcro. I kind of point to this and be like, oh yeah, we just highlight that. This is freaking cool stuff, okay? John Gilday takes an antibody from a rabbit, by the way, which is interesting. So the rabbit donates an antibody to ZO1 that we then tag with a jellyfish protein that glows in the dark. That's pretty damn cool science, all right? So that's just a little bit of cocky, like, hey, look at that. I just usually spin by this slide, and it just feels like I'm not giving John his credit. Miracle, under the microscope here, we are glowing with ZO1 protein. And what you see zippered together is millions of these small intestine cells that are making an intelligent, coherent barrier system in a petri dish now. If we add glyphosate at 20 parts per million, which is about the concentration you would see in conventionally grown beets and sweet potato, and some of my favorite foods on earth will have about that concentration of glyphosate. Within 16 minutes, you've blown apart the membrane. Destroyed the Velcro just in minutes. We're told by Monsanto and all the other chemical companies that make this product that it doesn't do any direct harm to humans because it just works through the shikimate pathway, which humans don't have, so it must be safer than water. And that was actually the first marketing campaign. Safer than water was the glyphosate campaign. We would argue that if you look at human cells within a system, it is anything but safe. It's blowing apart our self-identity at the cell level because right behind that membrane that just blew apart is your immune system. Every drink of water and every breath you take and every piece of food you're about to put in your mouth is about to hit your immune system. If you chronically do that, if you chronically overwhelm this gulf, this immune system with everything that you take in because you're now a leaky sieve, this is an inevitable graph. What we're looking at is the number of acres of wheat sprayed in black line here, the number of acres of wheat sprayed in the United States since 1990. It's been going up steeply in the last 2 decades. In yellow, you see the prevalence of the autoimmune disease celiac. We have lost the ability to recognize self when we start to build autoantibodies to ourself and to the gluten that causes a rheumatoid arthritis-like picture. And so we're getting huge immune dysfunction. And of course, it's here in the late 1990s where we start to see a huge prevalence of celiac and Crohn's disease. It's in the late 1990s and early 2000s that we start to see an amazing explosion of thyroid antibody disease in our young girls. By 2003, we did a study at the University of Virginia that for the first time went into elementary school just to to universally screen girls to find out if they were making antibodies to their thyroid gland yet. And astoundingly, by age 12, 25% of these girls were already making antibodies to their own thyroid gland. 1 in 4 girls destroying her thyroid gland by age 12. Her immune system is so confused about what's outside and what's inside, she's literally lost her self-identity at the molecular level, and her immune system is confused and attacking self. Extraordinary journey away from being human through killing our microbiome. This is what gluten sensitivity looks like under a microscope. I don't know if there's a switch handy back there, but if somebody sees a switch and you want to try flipping switches off, we can maybe see this better. Put me in the dark for a moment there. But again, we've got ZO1, it's lighting up the zipper between the cells, and then we've got the blue nuclei within the cells kind of showing you where the cells are. So here's zippered together cells. We add gliadin, which is the breakdown product of gluten, from the amount that you would see in about 1 slice of pizza, and it blows the membrane apart. This is gluten sensitivity or leaky gut under the microscope. And it turns out that this is very, very similar to the glyphosate process. And so we have control membrane, you add a little bit of gluten, and you get this drop in the protective membrane. You add a little bit of glyphosate, you get a drop in that protective membrane. But if you put those two chemicals in the same bite of food, a little bit of glyphosate and a little bit of gluten, you lose 80% of your barrier system. We started doing that in 1992. And so we started in 1992, and now most of the wheat in the United States is contaminated either directly or indirectly with glyphosate in the water system. 75% of the rainfall has glyphosate in it in the United States agricultural environment. 75% of the rain. 75% of the air is contaminated with glyphosate now. We are steeped in this stuff, and is interacting with proteins like the gluten protein that's been around since the beginning of time, never really causing us any harm until now. And so our group is about to publish this data showing that glyphosate is what accelerates the injury to gluten, and we just figured out how. So this is, again, John Gilday's genius here. But he figured out that glyphosate, when it touches the small intestine, upregulates CXCR3, which is a receptor for gliadin. The small intestine doesn't even really express a receptor for this compound until it sees glyphosate. And suddenly, you've got a membrane that's going to bind gliadin and create a leaky gut from gluten because of the predisposition or the injury that started with that glyphosate. As CXCR3 binds gliadin, it then triggers zonulin production. Zonulin now goes system-wide to destroy the Velcro everywhere. Not just in your gut lining create leaky gut, but leaky brain is instantly on the back end of this. I'll show you some of our data on that in a few minutes. So zonulin goes to create that leaky vascular, blood-brain barrier, leaky gut everywhere. What we're showing is the terahydrite, and I'll show you more on this in a few minutes. The bacteria in the microbiome can actually block that CXCR3 problem. And so if you have a healthy gut, you can eat gluten all day long and it's never going to cause leaky gut because you don't have CXCR3 receptors expressed enough to cause a problem. But if you're missing the bacteria and you've gotten antibiotics, you're now going to be triggering that CXCR3 response and getting injury. This is a quick reminder for me to tell you that there's more than Velcro between the cells. Also between the cells, so if we were going to look, this is a very tight on 2 cells that are abutted next to each other, and they're glued together by this black surface here. The tight junctions would be way out here, and as we zoom in under electron microscopy, we see another dense connection between these 2 cells. And if we zero in, it looks like a cable, a bunch of tiny little fibers, like a steel cable running from one cell to the next as an anchor point. But if we go even more microscopic and we increase the resolution, we find out they're hollow. Those are actually fiber optic cables running between 2 cells. Hundreds of thousands of those cables running from one cell to the next. Fiber optic cables. If you look even closer, at the very ends of those fiber optic cables are this. Perfect apertures, like you would see on a camera lens, that can open and close, letting light energy flow from one cell to the next. You are a light energy force. I think one of our speakers this afternoon is going to give us an earful on mitochondria. Just the theory. Mitochondria is one of my favorite things, and I hope we have a lively debate. He and I will have a debate from like 7 p.m. on tonight with drinks, I think, and we'll talk mitochondria. Mitochondria, he'll go on to tell you, create an enormous amount of energy, and that's light energy. It's electrical energy like you'd see from a nuclear plant. Surface of the sun compared to, one cubic centimeter of the surface of the sun compared to 1 cubic centimeter of mitochondria, 10,000 times more energy coming out of the mitochondria. Where is all that light energy going? It's going through the fiber optic channels from one cell to another to say, I'm Zach, 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 I am Zach. And that makes me powerful because I'm in one confluent fiber optic system saying, this is me, and I am in purpose, and I'm here on purpose, and I am okay, and I'm enough. All of these things. To be human is to be in communication with self in the light energy world. And so when you see glyphosate come along and blow apart a membrane, it didn't just blow apart the Velcro. It blew apart the fiber optic system. And you now have lonely cells that in just 16 minutes are turning into cancer cells. The predominant pathology feature under the microscope of a cancer cell is what we call the nuclear to cytoplasmic ratio. The nucleus within each cell should be a very small percentage of the overall volume of the cell. So a healthy cell has a lot of cytoplasm and a tiny little nucleus. As it turns into cancer, it loses the cytoplasm, and you get a high ratio of nucleus to cytoplasm, and that's a precancerous cell right there. You also see a shift from these nice globule round cells to these skinny oblong cells that are starting to look like fibroblasts. When you lyse the communication network, you immediately get a lonely cell, and within 16 minutes, we just took normal intestine to precancerous and cancer-type phenotypes in 16 minutes. And the World Health Organization finally said it's a probable carcinogen. And you saw the backlash the World Health Organization took for that little cautious step. California, God bless it for the frenetic insanity of California, but on the nice side of California, they finally went ahead and labeled it as a possible carcinogen in Prop 65 that just happened last year. Bizarrely, at the same time, they made it legal for the National Guard to move into a household to take a child away and forcibly vaccinate that child. So we have a little bit of a dichotomy happening in California right now. Not the smartest of public health systems we have going, but we are starting to recognize that glyphosate is a direct carcinogen and can quickly lyse the communication network between the systems. I don't need to go on to mention how unhealthy we are. We're 49th in the world right now. Croatia is way ahead of us in health outcomes. That's a war-torn nation that has no public health infrastructure whatsoever, and they have better health outcomes than the United States of America. Chile is kicking our ass. I mean, it's just like these developing countries are so much healthier than we are right now. So we got sick. We got sick through the exposure to antibiotics, through our farming industry, from our doctors and everything else, but isn't food supposed to be our medicine? Glyphosate does one more thing. It removes the medicine from your food. The shikimate pathway and these incredible things called plant plastids that live in the bacteria, the fungi, and the plants that grow within that soil, the plant plastids and the shikimate pathway make the alkaloids. And the alkaloids are the medicines within our food. We have anti-parasite compounds that control the behavior of parasites. We have anti-asthma compounds. We have anti-cancer compounds. We have cholinomimetics that would prevent things like attention deficit and autism, Alzheimer's. We have vasodilatory compounds that would prevent hypertension, which is now the #1 chronic disease worldwide. We would prevent with antiarrhythmics, cardiac arrhythmias and sudden death from heart attacks. We would prevent pain with the analgesics that are naturally occurring alkaloids within our food. We would treat and prevent diabetes with the antihyperglycemics in our food. And we did through all time until 1996. And we suddenly removed the medicines from our food chain with glyphosate. In that same decade, Monsanto, a chemical company that really started as a warfare company, they were providing most of the Agent Orange to Vietnam, would become the manufacturers of our food that would be purchased by our pharmaceutical companies. And that's just a perfect business model. Buy on the front end a company that grows 85% of the food staple crops for the country, that removes the nutrients and medicines and does damage to the species, all of the species that would consume that food, demanding a huge increase in my other company, which sells the pharmaceutical drugs back to the individual. It's an incredible journey. So I'm running out of time. Take a picture of that if you're taking pictures. This is glyphosate's legacy. I already told you all the information on this slide, but this is your kind of summing it up thing here. But of course, you saw this slide earlier. Mother Earth does reveal her grace. This picture taken by one of my best friends from medical school. He spent 6 months on the International Space Station recently. And it turns out that that's the Northern Lights from space. That's a halo over Mother Earth. It looks cool from down here when we see the Northern Lights. It looks like a halo when it happens in space. I think she is full of grace, and I think she is our greatest angel. Because in 2012, my group found that the very soil that we're killing with all of the glyphosate, 50 million years ago seemed to plan for this insanity. In fossil soils in the southwest United States, my group has been pulling through a cold water extraction process from a fossil layer of soil molecules made by the bacteria and fungi that are the antidote to Roundup. Weird, graceful planet that did that for us. She would predict that, and she would plan for that. And the microbiome 50 million years ago, we think of bacteria getting the fats, sugar, proteins, micronutrients out of our food, but what we found in 2012 was that this large family of molecules that are made by the bacteria and fungi looked a lot like the chemotherapy I used to make. And that was my aha moment. I was in clinic. White paper on soil science was brought in by William Vitalis, who's the unicorn. Walks in and says, Zach, I want you to look at this white paper. I'm pouring through it quickly, flipping pages literally as quick as I can because I'm late for a patient. And suddenly that's on page 42, and I stop cold. Goosebump moment, most important moment of my life probably, because I'm now here in purpose ever since that molecule hit my eyes. That molecule was in 2 dimensions, but what I saw was 3-dimensionally. This cluster right here looks a lot like the chemotherapy I made, and I said, oh my gosh. Here I'm running a nutrition clinic trying to reverse chronic disease through food, and I never realized that maybe all the medicine and the secret is in the soil. And when I found that that stuff was made by the bacteria and fungi, it made all of the alarms go off and all of the dots connect. This is how the microbiome and its shift is causing cancer in the human, is the anti-cancer compounds are being made by the bacteria and the fungi. And so that's been our journey now for the last 5 years. We've been studying this stuff intensively. And what we found is these snowflakes, each species of bacteria, each species, remember 5 million species of fungi that we know of today, makes its own subset of these molecules. And these molecules act as a wireless communication network to inform not just the bacteria and the microbiome and the balance between the parasites and the fungi and the viruses and the bacteria that must be in balance because they're letting us live here. If that amount of microbiome was against us, we would not be here. We're allowed to be here, and it's communicating across all of them. It turns out that it's also communicating across us. This is the control again. You add glyphosate, you blow apart the membrane. But if you add back the communication network to the microbiome, the whole damn thing zippers together in about 20 minutes. If you take the control membrane and you add the communication network beforehand and then try to attack it with glyphosate, nothing happens. No injury. In fact, we've now taken glyphosate to 20,000 times the concentrations you would see in the American diet, and we continue to see the microbiome intelligence continue to protect the system. Extraordinary journey. This is the article we published almost 18 months ago now on that ability of the microbiome communication network to protect against the glyphosate injury. This is what it looks like functionally. So I showed you the microscopy. You take a control membrane and you add back the microbial life. Look at the protein synthesis that happens within minutes. You almost double the amount of protection across the gut membrane with something from bacteria and fungi. No human input here. No bacteria or fungi, by the way. It's sterile. It's just their communication network. And you get this massive stimulation of protein synthesis of the extracellular matrix, tight junctions, gap junctions. You are making that cell more aware of what it is. If you add glyphosate, you destroy that awareness. But if you add the glyphosate and the microbiome back at the same time, no damage. That's small intestine on the right and colon on the right. Left, small intestine, right, colon. This is what it looks like as an antidote to gluten. Control membrane, add gliadin from the gluten, you have total membrane collapse. You add back the pterohydrate, everything zips back up again. The microbiome makes us intelligent. This is another journal article we published on reversing gluten sensitivity with the addition of that pterohydrate. This is what it looks like again. This is really cool. This is looking at the blood-brain barrier. If you add pterohydrate to the blood-brain barrier, you get near doubling of the barrier system between the brain and your peripheral nerves and the bloodstream. You suddenly go into a big protective state between the brain and the outside world. Interestingly, if you add gluten right to the blood-brain barrier, there's no damage. This is gluten here, gliadin. But this is really cool biology that John figured out. But if you add gluten to the small intestine, you damage the small intestine. I already showed you that. This is not the small intestine, though. This is the brain barrier with the material that's made by the small intestine after exposure to gluten. So what you're seeing here is gluten itself doesn't do the damage. You have to show the gluten to the small intestines. It makes zonulin that goes systemic and destroys the tight junction system. Incredible biology that's being teased out here to show that the main symptom of gluten sensitivity, and you've seen this if you have gluten sensitivity, it's not bloating and stuff like that. That may happen, but the primary symptom is brain fog. That's the hardest one to get rid of. You can get your gut a little better with digestive enzymes and whatever you're going to do, but this is hard to fix. And to see the microbiome intelligent enough to repair that in seconds, this is the microbiome now taking this damaged blood-brain barrier from gluten sensitivity being exposed to the small intestine, takes it way back, way past where it was at control. And so the microbiome makes us bulletproof to the most insane chemical results that we've ever played out. This is DPP4 enzymes. I don't have time to go into this because there's another one starting in a couple minutes. I'll give you more of this at the booth, but we've shown now that we upregulate the enzyme pathways that protect us. So let me just show you this slide because this is a summary. Gluten, glyphosate, bacterial overgrowth, all of that leads to toxic peptides. That makes zonulin in the small intestine, which goes to tight junction injury, the gut, brain, and the area, and you end up with chronic inflammation. But if you take the pterohydrate back into the equation, and you get the microbial information going, you upregulate the enzymes that tear down the toxic peptides and actually tear up zonulin before you ever get the tight junction injury to begin with. We also have shown this, which is that glutathione, your main antioxidant, when exposed to the bacterial intelligence, goes up by 800%, sustained. Your body continues to make glutathione. At baseline, you don't make much. You give it back the bacterial and fungal communication network, an 800% increase in your ability to make glutathione. If you take a supplement of glutathione, it may last in your bloodstream for 4 minutes. These measurements are taking 18 hours after exposure, still making 800% more. Beautiful story. Unparalleled safety and the rest. I'm going to give you a couple of last slides that are summary slides so you can take a picture of them. This is the microbiome's maintenance of the extracellular matrix. These are the things we've proved out. Some of these published, some of them are on their way to publication now. And so we've been able to show again and again that we support every level of intelligence at the barrier systems within the microbiome and its communication network, as well as within our own antioxidant systems, our own mitochondrial health, all the rest by adding back in the health of the microbiome. So in the future, as physicians, we are going to grow your medicine with you. Soil management can return the medical quality of their food. Mycelium and bacterial management, composting, simple permaculture and biodynamic farming, regenerative soil management can all bring us back to health over the decades to come, and that's our mission now. I hope you've been changed not just by my microRNA. but by the words that you've heard today, and you can't help but do something different now. You've got to mobilize this information. You need to get this out to the world. You need to start to really look at how are we going to become healthy. We need to go back to vaginal birthing. If born by C-section, you need to quickly swab that child with mom's vaginal mucus all over its body, inside its ears, inside its nose, inside its mouth, rectum. Get that child covered with mom's vaginal mucus if born by C-section. You will add decades of healthy life to that child by that one simple intervention. Breathe nature. Get outside and breathe. Get your own food. Grow your own food. Know your farmer. Touch the earth and food that grows in it. Stand barefoot in the grass. Nakedness gets total extra credit. Eat fermented foods daily. Get that back in. And ultimately just hug, kiss, and generally celebrate every human, animal, and plant that you can touch. And you're going to have humanity return to you. So last one, I just want to recognize my team real quick. I think somehow just sped by my team. There they are there. And so my whole team there's 55 of them now strong that work every single day to make all of this information known to you. And I'm just the tip of that spear. So thank you for your attention, your participation, and go on.